I'm going to share a lot of information with you today, and I mean a huge amount, and no apologies for that. There'll be no tests at the end, but I just want you to get a feeling for what we're doing in terms of disease modification and why it's important to do it. Someone has given me this title, Is Disease Modification in Parkinson's a Reality? So you could see that two ways. One, is it a reality now? The answer is no. Is it likely for the future? Yes. And I'm going to describe what we're doing to make it a reality. So currently Parkinson's patients have symptomatic therapies with many imperfections, and you will be well acquainted with that, but nothing to stop the year-on-year neurodegeneration. So what do we need? We need symptomatic medications that work better. We need disease-modifying medications that work. So neurodegeneration in Parkinson's patients go downhill maybe 5 or 7% per year. None of the symptomatic therapeutics are available. None of the dopaminergic agonists, none of the MAO inhibitors or anything else can slow that. So we're trying to stop that, so at least keep patients exactly where they are at the moment, maybe even a little better. So over 30 years, if you're going downhill at 5% a year, that's huge. So Cure Parkinson's Trust does a lot of preclinical work. We work in regenerative medicine, but we also have a very large clinical trials program, probably one of the largest in the world in its type, and that's what I want to talk about today. So the brief is to evaluate, prioritize, and repurpose existing and new developing medications that may have benefit in Parkinson's. So I've got a few busy slides, but I'm not going to read them out. I just want to give you the concept that one of the reasons why we started, because we knew that there was a calcium channel blocker called isradipine that is used for treating hypertension. If you're taking that, you're 30% less likely to get Parkinson's. If you're smoking, something similar, protection. If you've got high urate levels, you're less likely to get Parkinson's. And within the last year, we found that if you're on salbutamol inhalers for asthma, you're about 40% less likely to get Parkinson's. So there's some biochemistry going on of various different types. So the task is to find what therapeutics might firstly slow the development of Parkinson's, but also to treat Parkinson's in a disease-modifying way. And there's various ways we do that. We look at what's happening in other therapeutic areas. We use mass computational approaches. We do a lot of preclinical research. We screen maybe a couple of 100,000 drugs on a particular, known drugs on a particular target that we think is important for Parkinson's. And we set up an international committee to foster this. And I want to talk about what that committee is doing and what we have done in terms of our trials. So we get a large group of world experts together for a 2-day meeting, and they decide which of the drug candidates which we suggest to them should go ahead for clinical trials. So when we started, it was 2012, and these were the erudite members of this committee. It's gone up to 19 now. There's more biological targets have come in. We need experts in various areas. And typically these are neurologists that also have big research clinics. That's the sort of focus. We bring in other experts as well. So this, you see at the bottom, Camille Carroll has just joined the committee in the last year or so. So this is what we present to that committee, detailed appraisal of different approaches. We get deep into the science. We're trying to work at the cutting edge of the science and the medicine. And this is what the committee receives about a month or two before the international meeting, which is once a year. And then that's their start point to do their own due diligent research. And then they come together for 2 or 3 days, and then they decide what of those drugs should go into trial. So they look at about 25 drugs per year. And after we did that first committee meeting in 2012, we published that information in this paper. So we've not really published since, because a lot of Parkinson's patients looked over our shoulders and said, oh, well, if those world experts are putting this drug or that drug into trial, we'll just go and take it. So we had huge numbers of patients taking some of the drugs that were approved by the committee, which we told everyone about in this paper. So we've been pretty quiet since, because we're concerned that Parkinson's patients are taking some of these drugs without us knowing the safety in a Parkinson's population. And one or two of the drugs are quite potent, and I'll come on to those briefly later. So this is just the 2012 voting for the various compounds that we looked at in that year. So several of those have gone into trial. Bydureon is actually finished now. Liraglutide, lixisenatide, Velaparib, about halfway down. I'm going to talk about that in a minute. It isn't in trial yet, but we're going to get it into trial pretty soon. Nilotinib, just below it, is in trial, and that's a drug with a black box warning. It's used for leukemia, but there's compelling reasons to believe why it should be very good for Parkinson's disease. And we've now got a number of nilotinib-like compounds, and we're taking some of those into trial. But Deferoprone, just below it's in trial. Simvastatin, Camille Carroll is the PI of a 230-patient Simvastatin study. 2 years, patients are exposed to that drug. We might get a chance to talk about that a little bit later. So quite a few of those drugs got into trial. Now, we have actually published quite recently. We described the linked clinical trial initiative as it was in 2012. And we did that because our president, Tom Isaacs, recently died, and he had a huge hand in setting this up. So and there's a whole edition in a journal published actually very soon that is in tribute to Tom. So we did write this, and we talked about the various trials that came out of that very first meeting, and there's been effectively 10 of them. So what have we done in that time? So we've looked at about over 2,000 drugs. We've looked at more than 100 biological targets of relevance to Parkinson's, all addressing disease-modifying objectives. 173 drug dossiers have been considered actively we have a triage process, so the committee doesn't look at all 2,000, we're just the most promising ones. Of over 7 years, 49 drugs of the drug dossiers that we've discussed amongst the committee at that annual meeting have been prioritized to enter phase 2 trials in Parkinson's patients. And we're actually going to put 1 or maybe 2 into phase 3 trials fairly soon. So this is a sort of a timeline on some of our trials. So Bydureon at the top is now finished. It's a GLP-1 agonist. It's used in diabetes. It mimics a gut hormone, and there are brain receptors for this gut hormone, and that work was published in The Lancet. I'll come on and describe that in a few minutes. Simvastatin is ongoing, as I mentioned. Deferoprone is an iron chelator, which is important because Parkinson's patients have deposits of iron in the substantia nigra inappropriately, and there are iron chelators which are used for if you've given too much blood to a patient in a transfusion, you deal with that with an iron chelator. So these are also useful for taking iron out of the brain in Parkinson's patients. We've got 2 trials going on there. Ambroxol is and the one below is EPI-589, is looking at specific genetic forms of Parkinson's. Liraglutide is like Bydureon, it's a GLP-1 agonist. So I've got that in trial in Parkinson's patients who are insulin resistant in Cedars-Sinai Hospital in California. Lixisenatide 2 down is in Parkinson's patients newly diagnosed, like in 20 centers across France, 150 patients. So you can see there are different categories of Parkinson's patients. Disease-modifying therapy, you'd ideally give it to someone at the very beginning of their disease, so if you stop any progression, they're where they were at the beginning of the disease. But of course, we've got 10 million patients worldwide, and many patients are further down the line. So we're testing at different levels, and eventually we'll be moving into prodromal patients who have not yet got their motor symptoms, but perhaps have sleep disorders or lost their sense of smell or other indicators that they're on their way to getting Parkinson's. So we're managing, if you like, 40 or 50 years of the disease, trying to stop it developing wherever it is. So a whole bunch of different trials there. Nortriptyline is just about to start is centered in London Royal Free Hospital. UDCA above it is Oliver's trial. I don't know if will you get a chance to talk about that today? Excellent. Well, then I'll move on and look forward to that point. Azathioprine is a trial. It's an anti-inflammatory starting in Cambridge. Allagliptin, I'm launching that in a couple of months in Brisbane in Australia. So completed trials. Well, we've completed Bydureon. So that's moving into a phase 3 trial. So is disease-modifying treatments available for Parkinson's, which is sort of a paraphrase of the title of the talk. I said no, but after a phase 3 trial with Bydureon, it could be. So the results were strong enough in the Lancet when it was published that encouraged us to move to a phase 3 trial. Some of the patients responded exceptionally well. We were trying to understand what predisposes to be a responder and what predisposes to be a non-responder, but I think maybe 30 or 40% of the patients on that trial responded very well. So that's 3 to 4 million patients worldwide who could potentially benefit from Bydureon. So we'll do the phase 3 trial, and then that would be, if the results are good, then it will be available as a disease-modifying therapeutic for Parkinson's. And it will be the first one that will have that permission if it goes down that route. We've just finished an ambroxol trial in London, and the results are quite good for that, and I'm quite keen to get that into a phase 3 trial. Other ongoing trials, so I mentioned some of these already. Deferoprone trials are going on in various countries around the world, but focused on Lille in France, so that's where the PI is. I'm going to come on to EPI-589 and nilotinib in a minute. So these are the trials that we're sort of currently launching over the next year. So I mentioned the phase 3 Bydureon trial. So I just want to talk a little bit about I've thrown a lot of names of drugs, and I'm not, as I said, there are no tests, but I just thought I'd talk a little bit about the therapeutics themselves. So the GLP-1 agonists, so you'll see quite a few slides from our website. So if you want to explore this, you can go onto the CPT website. So we've got 3 GLP-1 agonists in trial, plus allagliptin, which is effectively doing the same thing, but by a different biochemical mechanism. So that will describe where this is coming from. So what actually happened was we were doing preclinical work 7 or 8 years ago, and I decided that it was ready to put it into Parkinson's patients, this drug called exenatide. It's the same as Bydureon, it's just a different formulation. And we published this paper on about 44 patients, I think, in 2013, and it encouraged us to do a much more comprehensive trial. We found that when we stayed with the patients for months 12 to 24, that they kept the benefit that they got in the first 12 months on therapy. So if you, so we got the controls. This is UPDRS at the top, and you'll see that that the patients on exenatide did quite well to separate out from the controls at 12 months, but the benefit that they gained over that year, they kept. They weren't on any therapeutic for the second year, but they stayed with that benefit that they had. Now this is just a pilot study. We switched to Bijoin. It's the same drug, exenatide. You can see it written on it, but it's a once a week rather than a twice a day injection. It's just a micro-injection, exenatide. And then this was published in The Lancet just over a year ago. So this is the follow-up study. We've written a lot about it, about how it might be delivered to Parkinson's patients. And the results were quite good. And as I said, some patients responded exceptionally well. And that's really what interests me, because let's work out what drug works for which patients and sort out the genetics and ways to determine that as quickly as possible so people get on the right treatment for them. So moving on to some of the other trials. So this is also the CPT website. This describes the deferoprone trial, the iron chelator trial. I'm hoping that you'll get copies of these slides available to you. So this just describes the structure of the trial, and if you're interested, you can look at that. We've got a really interesting drug called EPI-589, which has come out of pediatric mitochondrial treatment of patients with pediatric mitochondrial diseases. And we've been mitochondrial dysfunction is a major problem in Parkinson's. It's not the only problem, but it's certainly a major contributor to what's going on. So we've been running this trial using this drug which tackles mitochondrial function, something inside the mitochondria called oxidoreductases. Mitochondria are the parts inside a cell which produce energy to allow the cell to do whatever it needs to do, whether it's a skin cell or a brain cell. And also, fatigue is quite common in Parkinson's disease, and this is almost certainly mitochondrial related. So we've got 6 hospitals. This trial is now just about finished. And we've developed a very unusual biochemical readout as our primary objective, as our primary outcome. And this trial has also been done in parallel with ALS patients. And we have the biochemical results of those, and they're very good. And we're just probably in the next month going to get the biochemical results on the patients with 589. And we're going to expand this trial to many more hospitals around the world. Simvastatin. Camille, will you get a chance to talk about your Simvastatin study? Where are you? Oh, well, Camille is a PI of one of our trials, 230 patients, 2 years on Simvastatin. We published a paper last year suggesting the reasons why we think Simvastatin acts beneficially on the biochemical processes going on in Parkinson's. We suggested 7 different mechanisms. We're currently in the labs with an 8th. And when I get to Lille in 10 days, I think I will have the results of that, Camille. I think I will. We'll see. So Ambroxol, so there's a genetic group of Parkinson's patients with something called GBA mutations, glucocerebrosidase issue, and it's probably the largest cause of Parkinson's that's on a genetic basis. And there was a drug which was used for many years to tackle mucus in lungs. Who knew that it had this other effect on this particular enzyme? So That was discovered, and this enzyme is relevant for some patients with Parkinson's, particularly those who may be from an Ashkenazi or Sephardic Jew background. And so we've been doing that trial, and it looks like we're going to expand that trial. NAC is quite interesting. It's also mitochondrial, and I'll come back to that in a second. So about a month ago, we published a paper in Science, and so did Ted Dawson on a very interesting compound, well, a class of compounds called PARP inhibitors. And we looked at a PARP inhibitor every year in our annual meetings. The first one we looked at was Followup in 2012. And we now feel that it's exceptionally important going forward for Parkinson's. So we're trying to get a trial together as soon as we possibly can. So that was Ted Dawson's paper. Ted Dawson's a neurologist, scientist from Baltimore, Johns Hopkins, and he's on our committee, and he's published this astonishingly good paper. It's taken him 7 years, ever since about the time we first discussed it in 2012. And he's gone into the labs, and he's used various patient samples, and he's written a superb paper, which Patrick Brundin and I wrote the editorial on. And at one point, just up here on the right, we suggest that PARP inhibitors could be used with GLP-1 agonists, which I've already mentioned, in combination. I want to talk about combination therapies. So combination therapeutics really important because I'd be surprised if just one medication, we've got a lot of very strong biological targets, we're testing them, but I think that given that there's some pathology going on, you've got alpha-synuclein, Lewy bodies, that's causing a problem anatomically in the cell. You've got energy deficits, you've got inflammation. There are various targets to tackle at the same time. So I just want to leave you with this thought. So we've got the GLP-1 agonists, and most of those are in trial under our program. We've got mitochondrial compounds, so some of those are already in trial, some of them are about to start. And that's basically all energy homeostasis in green, and some other things going on. What I wanted to say was that alpha-synuclein, very different from energy homeostasis. They're quite different processes. So if you had two safe drugs, one tackling alpha-synuclein and one tackling energy deficits, then that's a beautiful combination therapy. Up at the top here on alpha-synuclein, we've got active and passive immunotherapies. So there's some companies with antibodies already in trial in Parkinson's patients. That should be published next year, so we'll learn more then. But we've got a number of compounds that we're working on as well. Our chairman, Patrick Brundin, just published showing how important the appendix is in terms of harboring dysfunctional alpha-synuclein, and tonsils as well. So this is an important area. Another important area, as I mentioned, is anti-inflammatory. So we've got a number of these compounds in trial or going into trial. Simvastatin affects a number of these different areas, including alpha-synuclein, which is not arrowed. And then we've got PARP inhibition, which I've just mentioned, c-Abl inhibition, lysosomal function. So how does this piece together in terms of the genetics? So we've got a number of genetic problems with Parkinson's, and this is how they interact with the pathways. What are we doing so far? So we've got a trial of EPI-589 studying patients with Parkin, pink, alpha-synuclein, various issues there. We've got Ambroxol looking at GBA patients. So if you like, this is the first step into personalized medicine. And also in different stages of the disease. It's an interesting time in terms of confining Parkinson's disease to the medical history books. We're not there yet. We're on the way. So the need for effective disease-modifying therapies for PD is urgent. We've launched a number of innovative and highly promising disease-modifying trials that have otherwise never been brought to the clinic. Thank you.
you.